Hello, everyone. I'm Yu Junlin, an associate professor in the Institute of Medical Medicine at NTHU. I would like to take this opportunity to introduce our latest work here. So it's about microtubule. Microtubule is one of the major cytoskeleton in living cells, which directly or indirectly regulate almost all kinds of cellular function and cellular architectures. So our understanding about microtubules mainly depends on the several microtubule disrupting agents such as nocardazole and colchicine and others. So people usually treat cells with nocardazole colchicine to disrupt the microtubule and to see what happened to the cells. This is a common strategy people has been widely used to study microtubule in the past 40 or 50 years. But the truth is those drugs do not work well. They actually disrupt the microtubule really, really slowly. So in these two cells, we labeled microtubule and treat them with the nocardazole and the colchicine. As you can see, within an hours, those drugs cannot efficiently disrupt those structures. So moreover, those drugs, they will freely diffuse to everywhere of the cells, which cannot target to the specific pool of the microtubule in living cells. So to address these long-standing issues, we try to develop new tools enabling us to rapidly disrupt the specific pool of the microtubule. So let me show you how fast of our systems. So we will start our system and treat cells with those drugs at the same time. As you can see, our system rapidly disrupt the microtubule and also completely disrupt the microtubule compared to those conventional methods. So how can we do that? So recently we have developed a series tool allowing us to rapidly translocate our protein of interest from the cytosol onto the microtubule by adding chemical compounds. So adding the chemical compound will rapidly translocate this protein from cytosol onto the microtubule within the 5 to 10 seconds, which is super fast. So with this tool, we try to translocate an engineered microtubule severing enzyme we call the microtubule erasers from the cytosol onto the microtubule, which will rapidly disrupt the microtubule, as you can see here. So translocation of enzyme that microtubule erasers onto the microtubule will not disrupt the microtubule, which serve as a negative control and indicating this event is enzyme-dependent manners. So now, beside the structural disruption, we also found disruption of those microtubule will also suppress the vascular dynamic in the cells. And before disruption, the microtubule, you can see those are lysosome. They move really fastly. They are super dynamic. But once we turn on our system to disrupt the microtubule, the lysosome become more static. They only can oscillate it locally. And beside the cytosolic microtubule, we can also target our system to the certain pool of the microtubule, for example, the promecilia. Promecilia is a microtubule-based structure protruding from the apical surface of the cells, which can receive the mechanical or chemical stimulation from the extracellular space. So we try to translocate our erasers onto these structures. As you can see, it will rapidly disrupt the promecilia within 5 to 10 minutes. To our knowledge, it's probably the fastest system to disrupt the primary cilia in living cells. Moreover, we can also target to other microtubule-based structure, for example, the intercellular bridge between the two dividing cells. So translocation of an eraser to the intercellular bridge will also disrupt these structures. We can also use the same pretty similar strategy to disrupt the mitotic spindle, as you can see here. Okay, and this system can be also controlled by the light illuminations. So we will turn on the light in these circle regions. As you can see, it's rapidly disrupt the microtubule only in the light illuminated regions. But once we turn off the light, the microtubule regrowth happens. So this system, control system is reversible. Okay, so with this ability to precisely control the microtubule disassembly in living cells, we have found several really cute phenotype that is depends on the microtubule. And also since several diseases like tumors and the neuron degeneration, they are a result from the microtubule defect. So we are also establishing the animal disease models. That depends on our strategy. Moreover, we also have developed several tools to precisely control the microtubule post-translational modifications in living cells, which we don't have time to go through the detail here. And beside the microtubule project, in the past 6 years, we also developed several tools using chemicals, light, and medical ultrasound to control cell activity. I will use one example to show you how we use medical ultrasounds to control cells. So we engineered an editorial sensing protein called prestin by introducing two mutant anti-NS, and found this protein can endow the human cells with a better ultrasound sensitivity. So a short pulse of the ultrasound stimulation can rapidly trigger the calcium response in the cell expression of the prestin anti-NS, but not in the control cells. We can also express the prestin antigens to the mouse brain regions and successfully use medical ultrasound to non-invasively activate those neurons locally. And also use this to treat the Parkinson's disease mice. So this graph shows the movement trajectory of the healthy mice and the Parkinson's disease mice. You can easily tell the defect of the movement in the Parkinson's disease mice. So after stimulation of the load by our system, it can greatly improve their movement defects. And it can also increase the key enzyme for the dopamine synthesis and the neuron growth factors. Okay, so this is the example. I quickly showed you how we use medical ultrasounds to control cell and also use to treat related diseases. So for the data I introduced today, I would like to thank the contribution from our lab members, and we have wonderful collaborators, and we are funded by the MOST and NTHU. Thank you for your attention.